Okay, I'm going to get started. Welcome to my note. My name's Esther Slomnicki. And today we're going to discuss age reversal, cancer cures, and winged wolves, exploring the possibilities of gene editing. I'd like to give a special shout out to my EP biology students, a couple of which have taken time out of their Sunday to come join us today, so thank you, and to my husband as well. Hi, Josh. So to start off, I'm going to talk to you and show you a trailer of a movie that came out this summer called Rampage. And we're going to watch a short 2-minute trailer, and then we'll find out what that has to do with our topic today. Hi, come on in.
I rescued George. He was 2 years old. He's not just a friend. He's family. Don't move a muscle.
Are you crazy? No! Don't move!
Okay. Very funny. You and I laughing at your joke. New guy, he no laugh. He cry. You can get up now.
And check your diaper. Come on. Let's go.
Hey, your message about George, you okay?
I don't know.
George? It's okay.
Is it me or is he considerably bigger?
No, he's definitely bigger. I need to find someone who knows exactly what the hell this thing is.
I found her. No, that's a lie. Technically, she found us.
What's happening to my friend?
Are you familiar with genetic editing? Changes will be incredibly unpredictable. Increased strength,
George!
speed, agility.
No!
Try to sales and chains they can't run.
Where's George? I need to see him.
He is dangerous.
I think it's time for me to turn the middle around.
We're gonna die!
Probably!
We've created the next chapter in natural selection. Project Rampage works.
You got another one of your little science experiments running around.
What do you mean another one? Incoming!
It's time to cross the line.
Of course, the wolf flies.
Stop it. Oh, somebody stop it. Stop it. Stop it. Please, somebody stop it. Stop it. Stop it. Oh, somebody stop it.
It's time we throw everything we can at these things and get people the hell out of that city.
Let's go save the world.
That's a big arm. Don't fight it.
You know there are other ways of dealing with people, right?
I do know that, yeah, but that's no fun. George? You okay? Ready to do this, buddy?
Okay, so in this movie, Dwayne Johnson's friend, George, was infected or injected with this gene editing tool called CRISPR. And as we saw, it had these unintended consequences of him getting bigger and stronger, and these other animals had developed these new traits of wolves flying and all these crazy animals. Is this our future? Is this science fiction? Or is this a possibility? Okay, by the end of our time together, we'll be able to answer that question. Okay, so just some background information about what a gene is, what DNA is, how do we do gene editing. So all living things have DNA inside of our cells, and our DNA gives us our traits. And specifically, this is one diagram of DNA. It has a shape called a double helix, this twisted ladder. I have a model actually right here that I can use, okay? And specifically, it's the middle part of the DNA, the nitrogenous bases here that give us our genes, our traits. How? There's 4 different types of nitrogenous bases we can have, 4 different letters, A, G, T, C. And it's the sequence or the order of these bases that code for specific traits. So the DNA is organized into sections called genes, and each gene codes for a trait, like eye color. All the DNA is wrapped very tightly into units called chromosomes, which are found in the nucleus of the cell, and each species has a unique number of chromosomes. Humans have 46 chromosomes. So we know a gene is this basic unit of DNA that codes for a trait, and you may hear the term genome. Genome is all of an individual's full set of its genetic information. And every cell in an individual has the same genome, except for our reproductive cells, the sperm and egg, and any cells that may get mutations during our lifetime. A mutation means a random genetic change. So you can actually isolate any cell from your body, and it'll tell you about your genes, your traits. And often the cells that are taken are cells that are most accessible, like from your blood or your saliva. And that'll give you a look into what genes you have. So currently there are genetic tests that can be done that will tell an individual about what genes they have, and the likelihood of passing down those genes or those traits to your offspring, your children. Can anyone think of any examples of a time in someone's life when they may have genetic testing done?
Before marriage.
Before marriage, that's a big one. Okay, why might someone want to do that before marriage?
You don't want two recessives to
Very
balance each
good.
other.
Very good. There are a whole bunch of diseases that are more common among Ashkenazi Jews, like Tay-Sachs and Gaucher. And if two people are carriers for this trait, that means that they have a 25% chance of having a child with that trait. So very often, women and men, before they get married, they undergo genetic testing to see if they're carriers for these traits. Any other ideas of when someone may get genetic testing done?
Diagnosis of cancer.
Excellent.
Rare diseases.
Right, very good. For certain diseases, you may want to get your genes checked to see if you have this disease because of a gene. Okay, when women are pregnant, sometimes they may get genetic testing done. Or has anyone seen advertisements for anything called 23andMe? Okay, right? So that's something that will sequence your entire genome and could tell you about your traits. Okay, so genetic testing is commonly done today. So I want you to take a minute and just imagine this scenario and think to yourself how you would feel about this. Imagine you've been offered a deal from a genomics company. You can get a free genome sequence, an analysis of all your DNA that includes a report of your ancestry, your traits, and a medical profile. The medical profile tells you about diseases for which you have a low risk of getting and also those you have a high risk of getting. Are you interested? Why or why not? And if anyone wants to share thoughts, I'm happy to hear it, or you could just think to yourself, would this be something that you would be interested in, knowing your whole sequence for any disease that you may have a chance of getting?
Well, there are lots of diseases for which there is no cure, so the question becomes,
what can you do? Right. What would be the purpose? Right. Okay. I see a lot of people kind of are shaking their heads like maybe not.
I think getting information is valuable.
Yeah.
What you do with it is something else.
Yeah.
And even if it's something that's incurable, you're a matter of passing that information on to your children and things like that.
Right.
And I think not to do it. I know halakhically we've had all kind of problems with BRCA positive women.
So I was hoping someone would bring up BRCA. Do you want to share? I don't know if everyone knows what BRCA is. Okay, BRCA is a gene that if someone has it, it makes them much more likely to get breast cancer. So if I'm sorry. Ovarian cancer, yes, thank you. So for that might be something that someone might want to find out. There is a steps you can take to try to prevent breast cancer or ovarian cancer if that's the case.
I would want to know who they share the information.
Very good. Okay, and that's a whole other topic of who has access to this information. Okay. All right. So let's keep this in the back of our minds because we're going to actually kind of take this even a step further soon. Okay, so now that we know what a genome is, what is genome editing? Genome editing allows scientists to make changes to a specific target site in the genome. It's almost like using a molecular scalpel to alter individual sections of your genetic code. And in this movie and in the news lately has been a very powerful new genetic editing tool called CRISPR. And we're going to go into detail soon about what is CRISPR, how it works, and what are the implications of using CRISPR. Okay, gene editing isn't new in the past 5-10 years. It's actually been done for many years. So a brief history of gene editing is in 1953, the double helix, the discovery of the double helix, this shape, was made by James Watson and Francis Crick. And with that knowledge came the knowledge that it was the sequence, the order of the bases in DNA that is crucial for determining your genes and your traits. So once they figured that out, they said, well, what happens if we try to modify this sequence? Change the order of the bases, what would happen? The first attempts to do this were just by causing random mutations to the genes, by exposing DNA to radiation or chemicals, and then seeing what the outcome of these mutations would be. All right, in 1973, the first genetically engineered organism was produced, a bacteria that had a gene for antibiotic resistance. From there, in 1982, the first genetically engineered human drug was made, insulin. Who's familiar? I'm sure all of us have heard of insulin before. It's used for people with what disease? Diabetes, okay? So people with diabetes need insulin. And before 1982, the way scientists and researchers would get insulin would be from other animals, okay, like pigs, or from cadavers, from human cadavers. So the problem is there aren't that many cadavers that are available, and pig insulin is not exactly the same as human insulin, so it doesn't work as effectively. So what they did with these, in creating this synthetic insulin, is they took the human gene for insulin, and they inserted it into a bacteria cell, and every time the bacteria would read and copy its DNA, it would also read the code for the human insulin, and it would produce human insulin in the bacteria. And then scientists could very easily just take out that human insulin and give it to humans. In the 1980s, that was the first attempt at gene therapy to try to fix a gene in an organism in order to either prevent or reverse a disease. It was effective in some cases, but it's complicated and limited. It really only works for diseases where there's a mutation in one gene. Okay, so like sickle cell anemia, which we'll talk about very soon, is something that gene editing would work for, and gene therapy. In 1994, the first genetically engineered food was made, something called the Flavor Saver tomato. And this was engineered to stay firm when ripe, but it actually bombed. It was expensive to produce, and people were very resistant to eating this tomato that was genetically engineered. However, since then, all right, there are many genetically modified crops. You may have heard of this because they're commonly referred to as, anyone know? GMOs, okay? And you may see on your food, oh, non-GMO. This does not contain any GMO. GMO is genetically modified organisms. So that's also a whole other topic of whether GMOs are safe or not safe, but it's very, very common. Corn is very commonly genetic, soybeans, exactly, canola, okay, all of these are things that are very commonly genetically modified. In 2012, this new gene editing tool called CRISPR is discovered. It's a gene editing tool that could be used anywhere on an organism's genome, in any living organism, humans, animals, plants. And like I said, we'll go into detail very soon about how this works and what this does. Okay, so let's talk in general about how gene editing works. So in order to modify a trait or cure a disease, we have to change a gene in the cells where that disease is found. Okay, so that could mean very few cells, or it could mean very many cells, which could be very difficult to do. We actually, in order to do gene editing, we're going to utilize a natural repair system that the cells already do. Okay, our DNA sometimes will break, and they'll break in a way sometimes which is called a double-stranded break, which would mean it would break right across here, separating the DNA from itself. So this happens during everybody's lifetime. So our cells have a built-in repair mechanism to try to fix the DNA, and it could repair it in one of two ways. Either it could take the two pieces and kind of get rid of some of the bases in the middle and then put the DNA back together, which is not very accurate, okay? It commonly leads to mutations. Or the cell can actually look for a normal copy of this gene. Would we have another copy of this gene? If we have a gene for eye color that got mutated, do we have another copy? Anyone know?
Any cell.
Yeah, okay. Actually, even within that cell, we actually do have two copies of every gene in our body, in our cells, because we come from two parents who are both contributing one copy of the same genes. So your cells actually should have a normal copy of that gene. So what the cells can do is they could find that normal copy, kind of copy over that regular piece, and put it in. Okay, and I'm going to show you a very short video of how the cell does that. Okay.
Can usually be fixed. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with 2 complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, change...
Okay, so we see, again, this illustrated how our DNA will naturally repair itself when there's a double-stranded break. Okay, so what if we can take this natural gene editing, not natural gene repair that our cells do, and use this to our benefit, okay? What if we can find a gene that is mutated, chop it up, make a double-stranded break, and we saw that the cell can find the normal copy of the gene, if it's present, copy it, and put it back in. So what if we can make sure that there is a normal copy of that gene there in the cell, find the mutated gene, cut it, and then just insert in that normal gene? That would solve a lot of problems. That would help cure diseases. Not every disease, but it could help cure diseases that are caused by a mutation in one gene. Okay, and this is really the basis behind how CRISPR works. Yes?
Now, would that damaged DNA be in one cell?
Okay, so there you go, okay? Yes, so this would only be in one cell, all right? If we want to cure a disease, we have to kind of insert the right gene in every cell that is caused by that disease, which is tricky, okay? So we're going to explore some possibilities of what we can do with this. Okay, so what is CRISPR? It's really called CRISPR-Cas9. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Okay, yeah. And Cas9 stands for CRISPR-associated. CRISPR-associated. I'm not sure what the 9 is. Okay, so this was discovered in 2012. Okay, and it was actually originally found in bacteria. It's this enzyme found in bacteria. Bacteria have, it was found that bacteria have these DNA sequences that are repeating sequences, but there are what we call spacers in between these repeating sequences. And scientists found, and they were quite surprised by this, that these spacers were actually copies of DNA from viruses. Okay, now a lot of times we kind of group bacteria and viruses into the same category because they could both cause diseases in us, but they're actually 2 very different things. Bacteria are living cells, viruses are not, and there are viruses that can infect humans and animals, and there are also viruses that can infect plants, and there are viruses that can infect bacteria and kill the bacteria. So what did the bacteria have? They have this immune system against the viruses that have infected them. If they were infected by these viruses and weren't killed by them, they actually took a piece of the viral DNA and inserted them into their own genome, okay? And next to those spacers was a code for an enzyme, Cas9, that would kind of take a copy of that viral DNA, a copy called RNA, and if that bacteria was ever infected by that same virus again, it has that DNA, that RNA actually match that's already there, and that tells that enzyme, chop up the viral DNA, okay, chop it up. And by cutting up that virus DNA, it makes the virus not able to replicate inside the bacteria. So the bacteria is well protected. Okay, so first of all, scientists were so surprised that something as simple like a bacteria had this this really complex immune system almost in it. Okay, and then they started to think, well, how can we utilize this to our benefit? The scientists who discovered this were named Jennifer Doudna, Emmanuel Charpentier, and colleagues, and it was refined by a scientist named Feng Zhang and colleagues. So if you want to see CRISPR in action, okay, this is not working, okay, but it was a short electron microscope video of the CRISPR moving and actually cutting the DNA, okay? But I am going to show you one more short video. So actually, before I show you, let me explain. So how can we use this? How can we use this? What can we do? Any ideas how we can utilize CRISPR to our benefit? Okay, what if instead of having that virus DNA inside the enzyme, and telling the enzyme, look for the virus. What if we put a different gene? What if we put a gene that we want to cut, a mutated gene, a copy of a mutated gene from humans, and say, let's cut up this mutated gene? And then what would that do? To cut the mutated gene, how would that help us? Any ideas? Yeah?
Well, isn't that what happened in China?
Oh, I'll get to that. China, the genetically,
Something like
the baby.
like sickle cell disease.
Yes.
You should be able to fix the.
Okay, good. By cutting a mutated gene and making sure that there's a normal gene there, like what we saw, the cell will just make a copy of the normal gene and put it in. But now we have like this system of finding the mutated gene. Okay, instead of just making random cuts, now using CRISPR, we can code for it to find the mutated gene among our over 3 billion bases. Okay, so let's watch how we do this.
Scientists have long imagined being able to make specific changes to the genetic code of a cell or organism. A revolutionary genome editing technology invented by scientists at UC Berkeley and Umeå University makes this possible using an RNA-guided protein called Cas9. Cas9 searches through the cell's vast genetic material until it finds a sequence of DNA that matches the sequence of its programmable guide RNA. The guide RNA enables Cas9 to open the DNA helix, then to position 2 molecular blades to cut each strand of the DNA double helix. Repair enzymes seal the gap in the broken DNA, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using this Cas9 technology for research applications in human health, agriculture, and bioenergy.
Okay, so we saw in this case that yellow sequence that was added in was a new DNA code that we supplied. Okay, but at the very least, what we could do for it with CRISPR-Cas9 is cut a gene, cut any gene we want. Okay, so let's revisit the thinking question I gave you before. Remember that genomic company? Okay, let's say for the first 100 volunteers, the company is offering to correct several of the disease-related genes found by the analysis. Imagine this were a very new procedure approved by the government for safety, but without a great deal of long-term study. Would you volunteer for this added service? Note, this service is not currently available and will not be in the near future, so use your imagination. However, I want to say that it may not be so far off, okay? Would you volunteer to have your disease-related genes corrected? Okay, that's just something to think about. Let's talk a little practically. Now that we have CRISPR, what can we do with CRISPR? So there's kind of 2 directions we can take with CRISPR. We can use it on either what we call our somatic cells, our body cells, and change the genes in cells that are in our body. And that would only change the individual itself, but not affect the future offspring of that individual. Or we can change what we call germ cells, the sperm and egg. And this would be modifying the genomes of not only that organism, but any offspring that come from that person. It'll change all future generations. And this second one is much more subject to concern and discussion by scientists and ethicists and the general public. Let's first focus on the somatic cells, the body cells. So the 2 categories of using CRISPR within our somatic cells are either to use what we call an ex vivo technique or an in vivo technique. Okay, in vivo means in the body, in the cells while they're still in the body. Ex vivo means out of the body. So for in vivo, we would have to insert CRISPR-Cas9 into cells while they're still in the body and change the cells while they're in the body. But that's really only useful for cells that are accessible, cells that we can reach, like cells in the liver. Okay, but ex vivo trials, we would take out the cells, change them, and then put them back in the body. So that would be useful for example, cancer, okay, or a blood disease where we could like sickle cell anemia, where we could take the cells that make blood out of the body, modify them, and then put these stem cells back in the body. Okay, so there are some trials on cancer using CRISPR, okay, and I'll just briefly mention 2 trials. where researchers took out the special white blood cells that are used as your immune system, called T cells. They took them out from cancer patients, and they used CRISPR to stop the cells from producing a protein called PD-1, programmed death 1. Okay, usually this protein prevents these white blood cells, these T cells, from attacking your own body cells and only attacking foreign cells. Okay, but by deactivating this protein, this makes the white blood cells attack your own mutated cells, so it's more effective in treating cancer. Results from this trial are mixed, and it's not something that's a treatment for cancer yet. Another trial that's been done using using CRISPR for cancer is for a specific cancer in liver and prostate cancer, specifically in these cancers that are caused by a chromosome rearrangement in these cells, where the chromosomes look different, the base sequence is different. So what scientists have done is they program CRISPR to detect that rearrangement, and when they detect it, they cut it there and insert a new gene that actually causes the cells to commit suicide. It's a cell suicide gene. So the cancer cells will kill themselves, okay? And again, this also is still in the early stages of trial. It is not yet commonly done, but there is research being done on curing cancer. Okay, we're going to focus on one specific disease called sickle cell anemia. Does anyone know anything about sickle cell anemia or sickle cell disease? Anyone know what? Yeah, go ahead.
It affects blacks. It actually protects the blacks in their own native environment from amoebic.
Malaria.
From malaria.
Yeah.
And it's a horrible disease.
Yeah.
If you ever see a young black kid who's had a stroke, chances are they had sickle cell disease that led to the stroke.
Right, okay. So sickle cell anemia is a blood cell.
It affects the oxygen carrier.
Very good. Yeah. It's a blood cell disease, specifically red blood cells. Okay, so typically we have a protein in our red blood cells called hemoglobin. And when the gene is normal, it makes hemoglobin that kind of really basically looks like this, these 4 chains that are stuck together. Okay? But in people with sickle cell anemia, there's just one base, one letter in the whole gene that is different. Okay? Instead of having an A in adenine, it's a T, a thymine. That one letter is enough to change the whole gene so that the hemoglobin clumps. The hemoglobin does not form properly, it clumps. And then the red blood cell doesn't form properly. It's a sickle-shaped cell, like the farmer's tool, a sickle. And then the red blood cells get clumped in the blood vessels. And as this gentleman mentioned, it's actually much more common amongst African Americans because it actually confers a resistance to malaria, which is a red blood cell disease passed on by mosquitoes. I could spend a lot of time talking about that. People with sickle cell anemia die young from sickle cell anemia. People who, in Africa, where malaria is common, people without sickle cell anemia may be more likely to die young from malaria. But people that are actually carriers for sickle cell anemia, they have some red blood cells that are sickle-shaped, and that's enough that they're not dying young from sickle cell anemia, and it makes them resistant to malaria. So this is something called a heterozygote advantage, like they're heterozygous, which means they have one normal copy and one not normal copy, but it's a selective advantage. So that's actually why it's more common in Africa. Okay. All right. So we have this mutation. How can we use CRISPR to solve this disease, to help cure this disease? There are two methods of doing this. We actually have 2 genes in our body for hemoglobin. One is called the adult hemoglobin gene, and that's the one that's mutated in people with sickle cell anemia. And we have another gene that's the fetal hemoglobin gene. When we are babies, we have a different hemoglobin gene that produces hemoglobin. And then very soon, while you're a baby, that fetal hemoglobin gene turns off, and the adult one turns on. Okay, and in people with sickle cell anemia, the fetal hemoglobin gene is not affected. So we have two directions we could take. Okay, either with CRISPR we can correct the adult hemoglobin gene. We could program CRISPR to cut the mutated gene. Cut the mutated gene and insert in a normal copy of the gene, thus allowing the adult to produce normal adult hemoglobin. Or we could deactivate the protein in people with sickle cell anemia that turns off the fetal hemoglobin gene, and that will keep the fetal hemoglobin gene functioning, producing normal fetal hemoglobin. And this second method has actually been much more promising. It's been tested in mice. Often these are done on animals, of of course, before humans. And as of October 2018, which was just a few months ago, the FDA gave the go-ahead to begin this trial on humans. Something to think about, though, is what could be a consequence if we eradicate sickle cell anemia? No, right, then malaria may end up being more common. Okay, we are getting rid of that resistance to malaria. So one thing kind of can lead to effects that we can't necessarily predict. Okay, let's focus on, yes, question?
Will preventing the fetal gene from being turned off prevent the adult gene from being turned on? Meaning, will they both be expressed?
No, so right, so it's turning, I believe it's turning off that gene that makes the switch.
Oh.
So it's kind of turning off the mutated adult gene and keeping on the fetal one. Okay, so now this has been really in the news, especially in the past month, using CRISPR on germline editing and the potential for having what some people call designer babies. Okay, so what is germline editing? Germline editing is, like I mentioned earlier, editing the germ cells, okay, the sperm and egg, or a very early, a zygote. A zygote is the cell that's formed from the sperm and egg joining together. It's one cell, and that zygote very quickly does mitosis. It divides to make multiple cells, a ball of cells called a blastula. This is all within the first couple days. And then very soon after, those cells start to change. They start to differentiate to form your tissues and organs. So in germline editing, we're introducing CRISPR either at that zygote stage, where it's one cell, or at that blastula stage, where it's a ball of cells. So in making that change in one cell, we are effectively changing all the cells that come from that cell. Okay? So this sounds like it has amazing things we could do with this, okay? If we change that one cell, right, that disease gene that we want to change will be gone forever in that organ. There's no worry, are we getting the right cells? Are we getting the right organs? We are changing we're getting rid of that disease from that person, okay? And actually, in 2015, there was the National Academy Summit on Gene Editing, and Harvard University geneticist Dan MacArthur tweeted the following statement. He said, prediction, my grandchildren will be embryo screened and germline edited. It won't, and he used quotes for this, it won't change what it means to be human. Rather, it'll just be like a vaccination. Okay, he's predicting that this is going to be just typically done almost as if it's like a vaccine. Risks, okay? Currently, it's not efficient and it's not specific and it could do more harm than good. If we make a change in that zygote, okay, and we do something wrong, the change isn't what we expect, okay, that mutation will be passed on to all of the cells of that individual and any subsequent generations that come from that individual. We can't even predict the long-term consequences of what will come, and we may not be able to reverse the effects. So it could have very, very dangerous consequences. So you may have seen this in the news. Just over a month ago, November 26, 2018, an article in the New York Times came out. Chinese scientists claims to use CRISPR to make the first genetically edited babies. Okay, so what's the story here? The scientist named He Jiankui altered a gene in embryos, leading to the birth of twin girls who he claims are resistant to HIV infection. All right, the way he did this was he disabled this gene called CCR5 that is used to make a protein that HIV needs in order to enter a cell. So by disabling the protein, HIV cannot enter the cells, okay? He did not publish any research in any journal, and he did not give evidence, okay, which is very uncommon amongst researchers when they make statements like this. This is actually illegal in the United States to do germline editing that actually leads to babies, and in many other countries. It's not illegal in China. It's opposed by many researchers there, but it's not illegal. However, after this statement, 122 Chinese scientists issued a statement calling Dr. He's actions crazy. And actually his research was shut down after this, okay? Dr. He said that the reason why he did this is because the father of these twins has HIV, and people with HIV in China face extreme discrimination, and now this father has a reason to live, knowing that he has children that are resistant to HIV. Okay, but the hospital believes this is a serious violation of academic ethics and academic norms. Why? The advisory group in China said, and really worldwide says, that genome editing should really only be done to address, and this is a quote, serious unmet needs in medical treatment. It had to be well monitored, it had to be well followed up, and full consent has to be in place. Is this a serious unmet need to make this baby resistant to HIV? It's not. There are actually much simpler ways of having children that will not get HIV, okay? The HIV is not in a sperm cell. It is if a man has HIV, the HIV virus will be released with the sperm cell. But a very easy technique is to simply wash the sperm cells and then inseminate the wife with this treated sperm cells to make sure that there are no HIV viruses. Or they can also do in vitro fertilization, where they take a sperm from the man and an egg from the woman and take one sperm, it's called ICSI, intracytoplasmic sperm injection, injecting that one sperm into the egg, and then you will have a baby that does not have HIV. So this is extremely controversial that he did this. Like we said, this really leads, going back to this picture, this leads to a slippery slope. All right, once we start changing the genes for diseases, all right, how can you make the line of, well, what's a disease? Okay, is obesity a disease? What about deafness? Okay, these are things that nobody would choose to have, but is this in the same category as sickle cell anemia, Huntington's disease? Yes.
Also, you said the way he did this was by disabling a certain protein.
Yes.
What if we learn later that
Excellent.
protein is essential in some other way,
Right.
or advantageous in certain other
Excellent,
ways?
excellent. Right. There aren't long-term studies of the consequences of this. We don't know. Yes, we know that will prevent HIV, but like you're mentioning, what if this will prevent other things that the babies need to happen? Okay, so it's extremely.
Also, there's been so much. I remember when I was a kid, which is a long time ago, and we're learning in school how many they recently cured polio,
Yes.
Created vaccines for polio.
Yeah.
And we're all saying, and there were other things, coming along. Like in those days, everybody got chicken pox.
Mm-hmm.
Everybody got the measles. And now it's like, oh God, somebody got the measles.
Right.
Like when I was a kid, well, everybody got
Right.
it.
So this actually,
But
yes.
everybody was thinking, well, we're going to cure all these diseases. And over the past 50, 60 years, we've done remarkable curing diseases, but new diseases have appeared that are really frightening.
Yeah.
Nobody had AIDS.
Yeah.
When I was a kid,
Right.
it didn't exist in the human population. So it really, you think you're so brilliant and we're protecting everybody, and
We don't know what's to come. We don't know what changing something.
You're not gonna have a disease-free world.
Right.
You're gonna have a kid with, you think, the best vision, the best, no baldness, wow.
Yeah.
Great IQ, but when he's growing up in a whole population where everybody's got fantastic IQs,
excellent.
great vision,
There's a fantastic movie
how great is,
right.
how much better is
Right.
it?
I show this movie to my AP biology students at the end of the year. It's a movie called Gattaca, Gattaca. It's actually named at it, like it's a nod to the fact that the bases of DNA are G-A-T and C. Gattaca is the name of a space station in this movie. It was actually made in 1998, which was a long time ago, but it deals with this idea of gene editing, and of course CRISPR was not even discovered back then, but it deals with the idea that this is our future where children are only made by choosing the best traits from each parent, okay? And it shows that the premise of this movie is there's this adult that is made the normal way, and he has this really high risk of heart disease, okay? And it shows this scene, this really powerful scene in the movie where the baby is born, he's born, and right away the nurse takes a blood sample and reads out likelihood of heart disease 99%, likelihood of depression 40%, likelihood of this X%, and then says anticipated age span 38 years. Okay, and this is told to the mother right after she gives birth. Okay, so in the movie, this boy grows up and he wants to be an astronaut, but instead of having a regular interview, basically, in order to get into this astronaut school, you have to have a genetic profile, and they said it's technically illegal, but it's done anyway, And anyone with genes that predispose them to certain diseases is not allowed to be an astronaut. So, I mean, the movie, it just, I love showing it to my AP Bio students. We just talk about, like, what is this, is who you are based on your genes or your motivation, your nature and nurture. Okay, we were just talking about this on Friday, right? But it can be our reality. This can be our reality.
And your likelihood doesn't mean that you're going to
Right.
be cursed with it. It's not
So
really-
actually in the movie, I mean, you should watch it, but he borrows DNA from somebody else and passes himself off as somebody else.
So like a urine switch test.
I mean, it's actually ridiculous what he does in the movie, but he ends up being a successful astronaut despite all of his genes, which told him that he can't be. Okay, so it's a fantastic movie. Okay, so this could be our future. This could be our future. There are many other questions that arise from this. What if we if we eradicate diseases and disabilities, will that remove some of the financial and emotional support that people that are still having disabilities encounter? Will that remove funding from certain disabilities? Are there any religious issues with doing this, with playing God almost?
There's always injuries. I mean, there's always going to be disabilities.
Yeah. The flip side to this, though, is how can we not alleviate the suffering? If we have these tools, okay, how can we not eliminate diseases? And we could say that we already kind of do in some ways. Very often women get tested when they're pregnant to see if their baby will have a high likelihood of having Down syndrome. And it's become more common in recent years for women to have an abortion, although plenty of women still don't. But there are women that are having abortions if they find that their babies do have Down syndrome or other severe genetic disorders. So we already kind of do have this selection of children for certain traits. So maybe this is just the logical next step. Okay. One more, and this is amazing. Okay, one more way we could use CRISPR is for organ donation. Okay, there's an incredible lack of available organ donors. So scientists are searching out different ways in order to allow organ donation to occur. So they found that actually pigs can be possible organ donors. Their organs are similar in size and structure to human organs. However, there's a big risk, a risk for immune rejection, that our bodies will fight off these pig organs because our immune system can recognize non-self cells, and also a risk for viral infection. The pigs have embedded in their genome certain pig viruses that can affect humans. Okay, so in October 2015, scientists used CRISPR on pig embryos to disable 62 viruses embedded in the pig genome. This was especially noteworthy because this was the largest amount of genes edited by CRISPR at one time. And they also altered 20 genes that could cause an immune response in humans. So therefore, this would essentially make this organ safe for humans to take in, okay? And they took it even further, okay? Which we're not there yet. But they thought, well, what if we can actually do this? What if we can actually use the pig to grow a human organ, a human kidney? How? We could disable the cells in a pig embryo, in a pig blastula, well, it's still just a ball of cells, disable the genes that code for the kidney to be formed, the pig kidney to be formed, and then we inject in some human cells into there, some human stem cells, and then what essentially should occur is these human cells would take over in the formation of the human kidney. Okay, so we have a pig with a human kidney inside of it, and then we could just take out the human kidney and use it for humans. And they did a trial of this in January 2017. They introduced human cells into a pig embryo during early development, and they found that indeed a human-pig hybrid embryo formed that had human muscle cells. Now, there were extreme regulations of how long they could let this embryo develop. It was only for a few weeks. They did not take it further. They didn't actually go all the way. Okay, but they did see that the pig embryo had the capability of forming human cells inside of it. And they were actually happy to see, and this was crazy, they were happy to see that there were no human nerve cells that formed, because if there were human nerve cells, that could spark human consciousness in a pig, which would be a very, very frightening thing to think about. So this would be the next step now, okay? But again, this is a little bit controversial. First of all, even just taking pig organs and using it for humans Could this lead to the harmful exploitation of animals? Will organs be available in a fair fashion to anyone who needs it? Will human bodies accept these organs long term? Okay, are there long-term consequences of having a pig organ inside of you? And the second study, certainly, Dr. David King, the director of Human Genetics Alert, said, I find these experiments disturbing. Okay, the formation of a human-pig hybrid embryo is a little bit of a disturbing thing to think about. Okay, CRISPR could be used in agriculture. Just to briefly name a few ways, we could change both plants and livestock. So wheat can be edited to be disease resistant. Potatoes could be edited to not sweeten on storage. Soy plants could be edited to produce healthier oils. Okay, dairy cows can be edited to not produce horns because their horns, sometimes they injure each other with their horns. Okay, we could edit pigs and sheep and cows to produce more muscle mass just by turning off one gene. All right. However, will there be societal resistance to this, like GMOs? Okay. Finally, and we touched on this in our discussions, the ethical implications and societal issues. Emmanuelle Doudna herself, who was one of the scientists who discovered CRISPR, advocates for restraint in the application of CRISPRs to humans until safety and ethical implications can be considered. Okay. However, other scientists do not feel that should be the case. They say full steam ahead. We can't withhold this new technology if we could potentially end human suffering. Currently, like I said, germline editing is illegal in the United States, in Canada, in many European countries. There's no federal funding in the US for this work. But in January 2017, researchers in the UK and Sweden and other countries have gotten approval to perform genome editing in human embryos just for research purposes. However, as we saw in China, they already took it a step further. All right. So we have to think of where should we use CRISPR? Okay. What should take our priority? Diseases, what diseases? Where can we draw the line to which diseases we should work on first? Once we start with diseases, does that naturally lead to things like deafness, short stature? And what about cosmetic changes? We don't know the long-term implications of what this can cause yet, okay? So is it safe to do human trials without knowing what the future will bring? In preparing for this session, I was amazed at how, it actually sometimes scared me to think about what our world could look like in 20 years from now using CRISPR. But it also enthralled me to think of what we could do using CRISPR. Okay, so let's just go back to our beginning trailer. The Winged Wolf, what do you think? Now that we know how CRISPR works, do you think it's possible? In the movie, I actually did not see the movie, but my understanding was that this wolf developed the wings he was already born, and then he was injected with CRISPR, and then he developed the wings. In order for that to be true, what did CRISPR have to do to this wolf? Any ideas? Yeah.
Add genes that weren't there.
Add genes that weren't there, okay, and a lot of cells, probably a lot of cells, okay? And this would also be assuming that there's only one gene that codes for the development of wings, which is probably not true, okay? So this is not likely to be reality now, or maybe far off in the future. But in order for this wolf to develop wings using CRISPR, we would have to modify many genes in many cells and have it suddenly have the ability to grow wings, which is not possible. Okay, but all the other things we discussed are not only possible, but they're our reality right now. Okay, so there's one video that I show my AP Bio students. If you Google, I don't even know, on YouTube, it's called CRISPR. It's about a 15-minute long video. It does not go into the science of CRISPR, but it talks about what we could do using CRISPR, and it's really a fascinating video to watch. I could find it after my session's done if any of you are interested in watching it. And also, if you need another, a movie for a movie night, I strongly recommend Gattaca also. So thank you for joining me today. If you have any other questions or comments, yeah.
This whole concept of